So I'd first like to thank the organizers for giving me the opportunity to talk about my research today. I am a postdoc in T.M. Murley's group at Virginia Tech, and together with Shiv Kale's group at the Virginia Bioinformatics Institute, we've been developing an algorithm to automatically reconstruct human signaling networks. So I don't need to introduce signaling pathways, and I'm in fact very glad that I am following the Cytoscape apps because what I'm going to be talking about are curated signaling pathways. So if we take the Wnt signaling pathway, this is the Wnt signaling pathway as represented by NetPath, one of many signaling pathway databases. Now, the interactions in this signaling pathway database are of very high quality, and that's because these are, again, manually curated. There's somebody going and actually annotating every single one of these interactions. Now, these signaling pathways are often highly incomplete, precisely for the same reason. Manual curation is a slow, tedious process, and we have lots of extra information about signaling pathways. So what we want to do, further, if you look at the Wnt signaling pathway in NetPath and compare it to KEGG or compare it to RECOMB, you see that there's actually very little overlap between these same representation of the same signaling pathway in different databases. So motivated by these challenges, we wanted to design an algorithm to try and automatically reconstruct these curated signaling pathways. So we can model these as directed graphs where the nodes are proteins and the edges represent interactions between proteins. And we're going to assume that signaling pathways start at a number of receptors and end at a number of transcription factors or transcriptional regulators. So if this is our curated pathway that we'd like to reconstruct, we want to do so in the context of a much larger, much messier protein-protein interactome. So this interactome represents all known interactions between human proteins. We can assume that this curated pathway, the pathway that we get from NetPath or KEGG, is a subnetwork of this interactome. And we can ask the question that if we remove the intermediate nodes and interactions, can we reconstruct this curated pathway? So our inputs to this problem are the receptors, the transcriptional regulators, and the human interactome. And I'm going to talk about an algorithm that we developed called PathLinker that returns what we call a proposed pathway reconstruction. So these are the intermediate nodes and the interactions between those nodes. Now, if you think about this problem formulation, it's very similar, in fact identical to a kind of a classic sources to targets graph theory problem. Right? We have two sets of nodes, and we simply want to find a network that connects them. There are many algorithms that do this, and many have been applied to signaling data. However, they tend to not perform very well in this pathway reconstruction problem. And so we have actually done a quantitative evaluation of these algorithms on a number of human signaling pathways from the NetPath signaling database. Finally, I'm going to talk about one of our results in the Wnt signaling pathway reconstruction and some experimental work there. Now, due to the time of the talk, I'm actually going to go a little bit out of order. I'm first going to talk about how do we evaluate a pathway reconstruction, and then describe the related work in terms of the performance. I'll then talk about PathLinker and provide some intuition about why PathLinker performs the way it does. So how do we evaluate reconstructed pathways? Well, we have at our disposal a curated pathway from NetPath or KEGG or RECOMB. And what we can do is we can layer on our proposed reconstruction. And from here, we can establish the true positive nodes and interactions, the false positive nodes and interactions, and the false negative nodes and edges, which are green, red, and gray here. Many algorithms, including PathLinker, often return a ranking of these nodes and interactions. And we can evaluate how well an algorithm performs based on precision and recall. Now for our positive set of interactions, we're going to take the interactions in the curated pathway database, the curated signaling pathway that we're interested in. Now, unfortunately for negatives, there's no set of interactions that are known to not be signaling interactions. So what we do is we subsample a set of negatives from the universe of all possible negative interactions, and we subsample at a ratio of 50 negative interactions to every positive interactions. In addition, we also exclude interactions that contain at least one protein that's in the known pathway. We call these kind of pathway-adjacent negatives. And these could be the fact that curators just haven't gotten to curating that interaction yet. So we constructed a protein-protein interactome from a variety of different data sources. And these directed edges include both bidirected physical interactions as well as directed signaling interactions. Now for our signaling pathways, we take 15 immune and cancer-related pathways from NetPath. And these are going to be the 15 pathways we're going to evaluate on. We establish our sets of receptors and transcriptional regulators for every single one of these pathways from previous studies. So we can compute the precision and recall of each one of these 15 signaling pathways separately, but I'm going to show you results where we look at somehow an aggregate evaluation. So we can apply the same algorithm to every single pathway and get a different ranking for every single pathway. We then combine these rankings into one really long list, and we can compute the precision and recall of that list. This is, in a sense, giving us an evaluation of how well these algorithms perform over a number of different signaling pathways. So I'm first going to start with some of the kind of most elegant solutions to this sources to targets problem. The first is simply, if we're going to find a subnetwork, let's just look for the shortest path. Let's compute the shortest path from every receptor to every transcriptional regulator. Okay. We can take the union of these paths as a single subnetwork, and that will correspond to a single point in our precision and recall curve. There are other algorithms. ANOT in particular has a really nice parameter that allows us to have a balanced trade-off between shortest paths and Steiner trees. And there's also been a lot of work on prize-collecting Steiner trees, and in this case, prize-collecting Steiner forests to solve similar problems. Now, each one of these returns a single point on our precision recall, and these are actually the best points after we tune a number of internal parameters. And what you see is while they achieve relatively high precision, they have very low recall, right? And this is actually intentional. The intuition behind these algorithms was to develop small, succinct subnetworks, right, that tended to be of high quality. So it's not surprising that they're going to end up here. Our goal is to kind of try and push this recall out. There are other algorithms, network flow algorithms, ResponseNet is one of them, that even does an even better job at precision, but returns very, very small subnetworks. IPA, the Ingenuity Pathway Analyzer, also has a network generation tool that we also implemented, and it returns dense subnetworks around a set of focus genes. And it's not necessarily connected, and in fact, in our results, we find that it's often not. But there is a parameter that allows us to start to push recall out further. However, the precision plummets. So we wanted to, push recall out and still maintain a reasonable high precision. So we had two different ideas. The first one was a k-shortest path approach. So instead of finding the shortest path between any receptor and any transcription factor, let's find some number k of them and take them as a unit. Okay? What this does is this allows us to push recall out, but and maintain relatively high precision in doing so. There's another very different idea that actually gave very similar results. This is a random walk idea, right? So we can think of executing a teleporting random walk where at every node a walker can teleport back to one of the initial receptors. So we're kind of restarting at the receptors multiple times. This gives you a weights where on the interactions interactions that are closer to the receptors have higher weight. It does pretty well too. So we had these two ideas, and it turns out that when we put them together, we do even better. Okay, so this, our algorithm, PathLinker, is simply performing a random walk on the network and then running a k-shortest path algorithm to find it. And it's striking that we get almost a doubling of precision at large values of recall with this method. So a little bit of detail about PathLinker. The first step, again, is a teleporting random walk that computes node visitation probabilities. Then we go from those node visitation probabilities to edge fluxes, edge scores on the edges. Then we use a k-shortest path algorithm to compute the k highest scoring paths. And then we can rank every interaction by the path that it first appears in. Right, to compute and compare it to our curated pathway to compute a precision recall curve. So the burning question is, why does PathLinker improve over other methods? And perhaps more importantly, why does it improve over its two individual components? There are two general reasons why. The first is that this K parameter is actually very useful. We can actually increase K to smoothly expand the solution and expand it in a way, an interpretable way. Right? We're just adding one more path. Further, while we are making this network larger and larger, we're still constraining that the paths must start at receptors and end at transcriptional regulators. So we're somehow keeping these paths close to the signaling pathway. But that can't be enough, because if you run KSP on this network, you get this pink line. The next thing, the second thing that's really crucial we found is that this first step, this random walk, is essentially weighting the network. Right? We're just taking the edges and we're weighting it according to how near they are to the receptors. And this very simple weighting scheme is the reason why we get this improvement in performance. We also weighted our network based on the evidence, the experimental evidence in these interactions, and we see an improvement in KSP on that weighted network. So weighting the network is really important. So up until now, I've been only showing you results on how we can reconstruct the interactions in a pathway. We can also do the exact same thing to reconstruct the proteins in the pathway, and it turns out that recovering proteins is a much, much easier problem. Okay, so and this is not just a path linker result. Every single algorithm that we ran can recover proteins very, very well. So really, it's the interactions in these signaling pathways that are a very difficult problem to try and infer. So this is the top 200 paths reported by PathLinker for the Wnt signaling pathway. I've colored the nodes. The pink nodes, and I don't know if you can see them, but there are some pink interactions, are those that are in NetPath. Nodes that are outlined in blue, and blue interactions are those that are in KEGG, another database, right? And the gray are those that are in neither. And what you can see is while there's some overlap in terms of the nodes between NetPath and KEGG, there are many blue interactions here that go to these KEGG-only proteins. It turns out that this is a non-canonical branch of Wnt signaling, the planar cell polarity pathway, that is present in KEGG but not in NetPath. So this is starting to get at the fact that there is surprisingly low overlap between KEGG and NetPath, and how these interactions, which would be false positives according to NetPath, are actually truly in Wnt signaling. So we looked at the first path that contained a protein that was neither in KEGG or NetPath. And it turns out to be this path from RIC to CFTR to DAB2. CFTR is a cystic fibrosis protein and had not been associated with Wnt signaling before. And it's important to see that even though CFTR and RIC are known to interact, and CFTR and DAB2 are known to interact, they're known to interact based on publications in cystic fibrosis studies and not in Wnt signaling. So we took this series of proteins and we experimentally investigated whether they are involved in Wnt signaling, whether CFTR in particular is involved in Wnt signaling. So we performed a number of siRNA experiments on HEC293 cells, and we used a luciferase reporter that measures the activity of TCF and LEF. And so the big takeaway message here is in the presence of Wnt3a, silencing CFTR reduces TCF and LEF activity compared to control, whereas in the absence, it actually slightly increases activity. Okay, so this means that there's something going on with CFTR and Wnt signaling, especially in Wnt3a signaling. We also measured cellular beta-catenin levels, where we see a decrease in CFTR upon the silencing of, a decrease in beta-catenin upon the silencing of CFTR. And we also conducted a number of co-IP experiments just to validate that CFTR indeed binds to RIC and DAB2 to verify those interactions. So to conclude, I've talked about PathLinker and how it can reconstruct proteins, and more importantly, it can actually do a pretty good job reconstructing the interactions of signaling pathways. I presented this quantitative evaluation of PathLinker and other algorithms on signaling pathways from NetPath, and I've shown some experiments about CFTR's involvement in Wnt signaling. There are a number of things that I didn't have time to talk about that we've done in the paper. We've assessed PathLinker on a weighted interactome, and we use PathLinker to bridge the differences between the NetPath and KEGG pathways. We also evaluated path linkers' robustness to noisy inputs, both in the sense of missing or extra receptors and transcriptional regulators, and missing evidence in human interactomes. Moving forward, what we're looking at is how we can experimentally define our starts and ends, right? Our receptors and transcription factors have come from curated signaling pathways so far. So we're working with the EPA to work with their ToxCast data, where they have screened thousands of toxicants and have protein response for those toxicants. I want to emphasize finally that PathLinker is really a hypothesis generation tool, right? People who are manually curating these pathways can use this as a guide to, can use it as a guide. And we've shown with this CFTR involvement that we can look for and experimentally verify novel proteins interactions in known signaling pathways. And with that, I'd like to thank people in the Morelli Research Group and the Shiv-Kale Research Group. The people in bold are co-authors of this paper, and this work is supported by NIH, NSF, and EPA. Thanks.
That was great, thank you. Are there questions?
Hi, thanks for a great talk. Since Gustavo's here, it seems like you've run a mini dream challenge. Maybe this is something that the community would be interested in tackling. And one thing I want to ask about is, given the motivation is that these curated databases are very limited, which I certainly won't argue, I think that's very true. Have you thought about other ways to do an evaluation that isn't dependent on the databases themselves, if you can bring in independent data?
That's a really good question. I haven't thought about an evaluation that does not include curated pathways. I think actually the next step for this evaluation would actually be to do something like take, for example, wiki pathways where there are many different representations, and somehow use kind of aggregate that information in some way to have more of a robust evaluation that's not specific on a single pathway. But yeah, I'd be really excited to talk to you more about that.
Thanks a lot.
All right, great, thanks again.